So we're really excited to have Blake Richards from MILA today. Blake did his undergraduate degree in cognitive science and AI at the University of Toronto. Then he went on to do his PhD with Colin Ackerman at Oxford, doing experimental work looking at neuroplasticity in tadpoles. Since his PhD, he has moved towards more theoretical work, essentially using neural networks as a laboratory for neuroscience research, which I think is what he's going to be talking about today. So yeah, take it away.
Thanks a lot, Augustine. So hi, everyone. Pleasure to be here with you today. Feel free to interrupt me during the talk to ask questions. I'm happy to pick myself up again after addressing anything. So let's keep this informal. So today I'm going to be talking to you about the functional specialization of visual cortex, and in particular how that can emerge from the training of parallel pathways with a self-supervised loss. Ultimately, the goal of this research is in part to understand the brain and how it is shaped, what loss functions shape it. But I think it also has some interesting implications for more kind of the engineering side of AI and thinking about how best to design AI systems, which is what I'll end on with a thought with that. So to begin with, for those of you who do not know any neuroscience, let me just give you a bit of a background primer. So within the mammalian neocortex, the neocortex being the part of the brain that is wrapped around your forebrain and which is responsible for most of your quote-unquote higher order thought, certainly all the stuff that makes us uniquely human. The neocortex has two primary visual pathways, which we refer to in neuroscience and psychology as the what and where pathways. So roughly, your primary visual cortex is at the back of your brains in your occipital lobe here. And so this is where retinal information will first arrive in the neocortex. And then it is ultimately kind of farmed out to two separate pathways. One which runs along the bottom of the brain, the ventral stream, which is more concerned with the question of what things are present and what do they mean. So for example, in the ventral stream, you'll find cells that are selective for faces or particular categories of objects, things like that. In contrast, there's another pathway that heads out along the top of your brain called the dorsal stream. And the dorsal stream is more concerned with the question of where are things located and what are they moving to? So, or where are they moving to, I should say. So what you find in the dorsal stream is neurons that don't really care about, say, the categories of objects or anything like that, but instead are very concerned with the direction in which things are moving and whether or not there's coherent motion in particular spots, things like that. And we don't, within neuroscience, fully understand exactly why these two pathways exist and what led, either through evolution or during the course of early development, what leads to their distinct functional roles. And I should say, you also see not only that these cells have these different selectivities, but lesions to the brain have different effects for these different pathways. So if you lesion the ventral stream, people will have trouble recognizing objects or people they know, things like that. Whereas if you lesion the dorsal stream, people have trouble, successfully grasping objects or moving out of the way of things, stuff like that. So why does the brain have these two distinct specialized pathways? Well, one of the things that we had been discussing in my lab, and which we can call kind of our hypothesis for why this might be the case, a sort of normative hypothesis for why this might be the case, is that visual prediction requires two different, potentially competing forms of invariance. So let's say you see some object, like a car, and it's moving in your visual field. In order to predict what you're going to see in the future, in the near future, you need to know two things. The first is that objects are permanent in their identity, and so you ultimately, if you see a car in this particular instant in time, chances are you're going to see an orange car in the near future as well, unless you completely turn away. So assuming that generally your eyes are, you're not constantly spinning yourself around. If you see an orange car, you'll probably see an orange car in a moment. And it doesn't really matter what direction of movement the orange car is engaged in, because as long as we're talking about a short time into the future, so literally just a few, like maybe 100 or 200 milliseconds, a second at most, whether it's moving left or right, you're still going to see an orange car in the near future. So you can make a movement invariant prediction about the presence of this category of object being in your visual frame in the near future. However, in contrast, there's an alternative prediction that you can make that is invariant to the identity of the object. If there is some object moving in a particular direction in your visual field, then regardless of what the category or identity of that object is, you can predict that the leftward movement of that object, say, is going to lead to something being in the left part of your visual field, for example, in the near future. So this would be object invariant prediction regarding movement. There will be leftward movement, and you know that regardless of whether it's a car or a dog or a ball, etc. So we have these two forms of invariance potentially to do prediction, and what we hypothesized is that the what and where pathways are specialized this way precisely because they help us to do prediction, and they have been shaped by predictive learning functions in the brain. There's a whole large body of work showing that the brain is concerned with prediction. I'm not going to overview that for you today, but I'll just say that although there's still work to be done, this is maybe settling down to be one of the known facts of the brain, is that it does seem to be concerned with predicting the future. And so the idea is that these what versus where pathways result from the predict the future due to these two competing invariances that I mentioned. And the way we wanted to test this hypothesis was by training artificial neural networks with a self-supervised predictive loss, and then compare to real neural representations in the what and where pathways, and ultimately determine whether or not there is evidence for that kind of specialization really emerging naturally from this predictive loss. So just as a preview of what I'm ultimately going to tell you, indeed self-supervised learning, but not supervised learning, is able to generate the specialized pathways that we see in visual cortex. And we've got a paper out on bioRxiv. If you're interested, you can go check it out. By first author is Shahab Bakhtiari. Okay. So how we're gonna, I've already kind of described how we're gonna test this, but in order to compare to brain data, we need brain data. And one of the great things about being a theoretical neuroscientist slash machine learning researcher nowadays is that there's lots of data available out there for you to use. And one example is if you need neuroscience data, the Allen Brain Observatory has a lot of open data sets available. So the Allen Institute for Brain Science has this program, the Allen Brain Observatory, where they collect a lot of data, in this case from mice, from many different brain regions, and then share the data openly. So in this case, we're using a data set which is described in this paper by De Vries et al. And briefly, if you're unfamiliar with how these systems work, they're doing what's called two-photon calcium imaging. That is a system of recording that allows you to record the activity of hundreds of neurons at once in an awake behaving animal that you're showing stimuli to. So after prepping the animal for these recordings and doing some intrinsic imaging to check out where they are in the brain and habituating the animal, they do two-photon imaging recordings. So they get the activity of hundreds of neurons in response to various visual stimuli. And in this case, one of the sets of visual stimuli that they had were quote unquote natural movies, basically just movies but not artificially generated stimuli. So, you know, movie clips like the one shown on the top right here. And then they imaged in the animal's visual cortex in a variety of areas, 5 different areas in total. Primary visual cortex, V1 or VisP, depending on who you're talking to, LM, AL, RL, AM, and PM. I won't go through the anatomical names of all these regions, but just so you know, they're judging these, they're doing recordings in all these different regions in response to these natural movies. And in order to compare neural networks to the neural data, what we're going to do is we're going to use something called representation similarity analysis. So it's pretty easy to understand. Basically, what happens is you've got your animal watching your movie, and so for each frame of the movie, I should just say, I'm going to describe the analysis we do here. RSA can be applied to any other stimuli. It doesn't need to be applied to just movies, but I'll describe it for how we apply it to these movies. So the animal's watching a movie, and what you get is over time, there are responses to each movie frame. You can calculate some lag for how long it takes for visual information to propagate in the visual cortex. And so at that lag, you record the calcium responses in the neurons. And then you're going to get that for different movie. So within n-dimensional space, you've got this trajectory through n-dimensional space, basically, that represents the responses to each of the frames of the movie. And that's going to give you a matrix where you have the number of columns equal to the number of neurons that you've recorded from, and a number of frames, number of rows equal to the number of frames. And then you just square that matrix, and this gives you what's called the representational similarity matrix. So this, of course, is going to measure. You can also normalize this just to make sure that it's not just measuring overall level of activity. And what it's going to measure then is basically how similar are the responses to the different frames. So each entry in this matrix now is going to be, so it's number of frames by number of frames. So each entry in this matrix represents, for a pair of frames, how similar were the responses across the population of neurons to that pair of frames. And you can do this both for real brain data and for an artificial neural network. If you play a movie to an artificial neural network, you can construct a similar representational similarity matrix. And then you can take your representational similarity matrices either between animals or between artificial neural networks, or between an artificial neural network and an animal, and you can compare. And this will tell you roughly something about the geometry, effectively, of the representations in that circuit. So to what extent are different frames being like having similar distances in space or angles between their vectors, etc., depending on the similarity metric you use here. So first, just as a kind of sanity check, we do RSA on the actual mouse data itself. So in this case, we're going to consider 5 regions. So we're going to leave out one of the regions that was in the Allen brain set called called RL. We left that region out, I'll just say, because it's a multimodal region, so it's only kind of partially driven by visual stimuli. It likes to have both vision and other stuff. And since this was just a visual data set, it wasn't giving robust responses. So we took the rest of the 5 regions, and as an initial sanity check, we did RSA on those 5 regions across mice. So what you're looking at here are the correlations between the representational similarity matrices between mice for different areas, because each mouse they recorded from a different area. And what you can see is that, which is nice, what you'd expect, is that the highest values for the RSM similarity sit along the diagonal, meaning that when you're comparing across mice, primary visual cortex is most similar to primary visual cortex in other animals. LM is most similar to LM in other animals, etc. So it does seem to be reflecting something real. This analysis seems to be reflecting something real about the representational structures in these different regions, because it says these different regions are distinct from one another, and across animals, the same regions are most similar to each other. Now, two other bits of analysis that we do to prep our work here is we also assess the hierarchical score for each region. So basically, depending upon a variety of anatomical measures, you can judge how high up the visual hierarchy the different regions are. This is basically, this is based on an analysis of kind of how many synaptic jumps has to occur for visual information to get to those regions, and also other factors like how much do these regions seem to be receiving feed-forward projections versus sending feedback projections, things like that. And there's a variety of different ways you can measure that in the anatomy. But this gives us for each region a hierarchical score, which indicating where it is in the hierarchy, sorry. And so at the bottom, we have primary visual cortex, which makes sense. That is where the retinal information first arrives. And then as you go up, you go through LM, AL, PM, AM. But it should be said, this hierarchy doesn't mean that these regions are feed forward to each other. In fact, what happens in terms of the anatomy here is that primary visual cortex will send out projections to all these regions, and they also connect to one another. So this is a soft hierarchy score. It doesn't reflect a pure feed-forward hierarchy, just so you're aware. And then additionally, what we do is we score each region on its ventralness versus dorsalness. And this is based upon both functional and anatomical data, and effectively what we get is that region LM is the most ventral-like for the mouse. I put scare quotes around this only because mouse anatomy is different from primate anatomy. So it's a little bit funny to me to use the words ventral and dorsal because that doesn't map fully onto the anatomy of mouse, but it's the functional pathways still exist. So this region LM is the most ventral-like, meaning that when people do recordings in here, and when you look at where it projects out to, it's similar to the ventral regions of primate brain. It has cells that respond to more categorical-like things, though not the sorts of abstract categories that primate brains do, it should be said. And it projects out to the sorts of regions that primate ventral areas usually do. Whereas in the other extreme, we have region AM here, which projects out to the areas of the brain that the dorsal parts of the primate brain do. So motor cortex, things like that, concerned with controlling movement, and which as well, based on the functional measurements, indicate that this region is interested in movement and the sorts of things that dorsal pathway is interested in. So we lay out the brain regions like this from ventral to dorsal, and roughly we're gonna make a cut here. We're gonna call these three at the most ventral ones are sort of ventral pathway, and the most dorsal ones are dorsal pathway. But it's worth noting that there's a gradient here, and the most extreme comparison is between AM and LM. Okay. Now, last bit of preparation before I can actually show you the results. How do we actually train our networks? So we're training with, in this case, the contrastive predictive coding loss function. And we take the approach taken by Hanadal in their paper on using CPC to learn from videos. So what we've got is a convnet that receives segments of the videos, and the output from that convnet projects into a recurrent neural network that is used to generate a context variable. That context variable is then used to generate a prediction for the next upcoming time steps as to what the output from the convnet is going to be. So I'm sure most of you are familiar with kind of modern self-supervised learning techniques, but just for those of you who are maybe more familiar with more traditional approaches in unsupervised learning, like generative models, an important distinction here is that we're not actually learning a generative model over the pixel data. What we're doing is we're just trying to predict the latent representations that come out of the convnet. And now what you have to do when you're doing that, though, when you're doing your predictions in latent space, is you need some way of preventing representational collapse, because obviously a trivial solution for this is to simply say, okay, well, get your convnet to output the exact same thing for every frame, and then it's very easy to predict what the next time step is going to be. So to prevent that, contrastive predictive coding uses what we call positive and negative samples. So what happens is you define your positive samples as the next frames in the movie. So those are the ones you're going to predict. And then your negative samples are the other frames from. And in your loss function, what you're doing is you're minimizing this, which is the sum across your pairs of the logarithmic ratio between e to the similarity, it can be dot product or other similarity scores of your positive pairs divided by the similarity of your negative pairs. So that's going to encourage your system to develop predictions, z hats here, that are as similar as possible to the real next frames in the movies, while being as dissimilar as possible to the other frames from the batch. And in this way, the system learns a non-trivial prediction for the upcoming latent representations. Now, the training data that we used in this case was the UCF 101 data set. And so this is, if you haven't seen it before, just a series of videos of different activities. Now, this is actually labeled data, and this is important for us because we're going to also do some supervised learning with the labels as a control. Basically, it's different activities with a label as to what the activity is. Here, lots of different kind of physical activities, sports, etc. And so you can either train off of these in a unsupervised or self-supervised way, just using the videos, or you can train in a supervised way by training the system to recognize, you know, this is swinging, this is diving, this is kicking, etc. And then lastly, what we do with our neural networks to run some additional kind of control comparisons is we have two architectures, which I'll refer to as ResNet 1P for one pathway or ResNet 2P for two pathways. So the ResNet 1P pathway is basically the same as from the original hand paper. It's a ResNet with this recurrent neural network on top that's generating your contextual predictions. But in the 2P pathway, after your initial convolution, you then split into two separate parallel pathways, which I'll always color code with red and blue here, before merging in the final layer, which is where you're going to make your predictions. And the important thing I'll just note is that to make these comparisons valid, we have the number of kernels in the parallel pathway architecture in a single pathway. So the number of total kernels across the pathways is the same, and they both have kind of equivalent computational capacity. The difference is that in this architecture, the comms are separated out into these two separate pathways. Okay, so I'll move on to the results now. Are there any questions before I continue? I'll take that as a no, so let's go. Okay, so one of the things we observed is that when you train the single pathway architecture, you end up developing ventral-like representations. So what I'm plotting here is on the x-axis, the layers of the ResNet, and on the y-axis, the RSM similarity. Now, I should say, so it says noise corrected here. So just so you know, all of these values are plotted as a percentage of the noise ceiling, where the noise ceiling is what we calculate by comparing an animal to itself. So obviously, the best you could ever do in terms of representational similarity is by comparing an individual to themselves, but they will have some noise in their own, like there's going to be some entropy to the distribution of neural responses. And as a result, there's going to be some maximum correlation that you can achieve in your RSMs, even when comparing the identical individual to themselves. So all of these RSM values are are normalized to be a percentage of the noise ceiling for a mouse for these different regions. So what we find is, and so really you can think of this as being the percentage of, total possible variance you could explain with these representations. So what we find is that when we look at the ventral pathways in the animal's brain, we see that as you go through the layers of the ResNets, you get an increase in the representational similarity. And in fact, that reflects roughly the hierarchy of the different regions. I'll discuss that more, but basically, primary visual cortex, we probably should have ordered this differently, but we've ordered it from most ventral to least ventral. Primary visual cortex, which is at the bottom of the hierarchy, has kind of the best correlations earlier in the network, whereas LM and PM have correlations later. They, clock in at around 60 to 70% of explainable variance. Whereas what we see in the dorsal pathways is that when we make that comparison, in fact, as you move through the neural network, so let me just actually clarify one other thing. So the gray circle here is if you do representational similarity analysis with the movie frames themselves. So that's pixel level correlation. So what we consider a kind of trivial level of correlation to some extent, or not trivial, I suppose, because you can always remove correlations as we show here, but to give you a sense of what like a very simple baseline is. And so what we find is that you only get a little bit better than pixel level correlations in AL, and in fact, you basically get worse pretty much throughout the network for AM in terms of the percentage of variance you can explain in the representational geometry, suggesting that what's happening in this ResNet is that the representations across the convolutional stack are becoming more ventral-like and less dorsal-like. This is just now a quantification of that across groups and also comparing to some other networks. So in addition to training with contrastive predictive coding, we also trained a ResNet just on object categorization, so ImageNet. We also had an untrained ResNet, but with the weights, so basically just shuffling the weights of the trained one, in fact, so that we keep the same statistics and weight strengths, but remove the information in the structure of the weights. And then a very simple model of 3D Gabors, which is like kind of the most basic model of visual cortex that you can have in neuroscience. And what you can see is that for the three ventral areas, VIS-LM, VIS-P, VIS-PM, the CPC trained net, so what we're plotting here is the max RSM similarity, so that is the maximum across all the layers. What you can see is that the CPC trained net does much better than a network trained on ImageNet. It does, and way better than an untrained network or just the basic 3D Gabor model. However, for the ventral areas, what we find is that CPC only does a little bit better for AL than an ImageNet trained network or the untrained network. And in fact, for AM, it's basically a little bit worse than the untrained network, in fact. So training with CPC led to worse representational similarity to this most dorsal of regions than if we had just taken a randomly initialized ConvNet, or ResNet, I should say. And then this is just looking at that hierarchy index for the different regions. So here it is laid out by hierarchy. So the hierarchy indices that we got for these, like where here what we're plotting is where did the peak occur across the layers. And what you can see, as I mentioned already, is that primary visual cortex is lower in the hierarchy, which is what you'd expect given the anatomical hierarchy score. Now, what's interesting though is that when we train with our two pathway model, now what we find is that in one of the pathways, which we color code blue here, we again get good fits to the ventral pathways. So the ventral areas, I should say. So, you know, as before, we find that there is an increase as you move through the network in representational similarity for LM, VisP, and VisPM. In this blue pathway, it roughly reflects the hierarchy to some extent. And we're getting similar sorts of values as we got before in terms of the explainable variance. But what's interesting now is for the dorsal pathway, we no longer get this drop in representational similarity for VisAL. It maintains just a little bit above the sort of pixel level. I would note though that this scale is kind of bigger because we're also dealing with the fact that in the blue pathway, you again get this drop in similarity to the dorsal pathway. But in the red pathway, it preserves its similarity more. And in fact, for VisAM, it actually gets better and better at matching the region as you move up through the layers of the neural network. So now when you do this analysis where you say, okay, what is the maximum similarity of the network, where you get a peak here in the representational similarity score. And here we're comparing CPC1 pathway to CPC2 pathway to the untrained network. What you find is that the ability to do a better match to the ventral regions than the untrained is preserved, because here we're considering both the blue and the red pathways, as well as for region AL. But now for this very dorsal region, region AM, we get a much better fit with CPC than we did with an untrained network, whereas with the one pathway model, we just get the same level. And then looking at the hierarchy index, we now also get an appropriate hierarchy. VisAM is higher up the hierarchy than VisAL, and so we get our peak higher in the neural network. So this is just another way of kind of looking at that and understanding what's happening in the neural network. So here we're plotting the representational similarity matrix similarity across the different layers of the two pathways in the two pathway architecture. And what you can see is that if you compare the red pathway to the red pathway, you get high values of representational similarity. Whereas if you compare the red pathway to the blue pathway, you do not. You only get a few high values out towards the edges of the network, but throughout the core of the network, it's very low similarity values, showing that indeed you have a specialization in terms of the representational geometry. So the two different pathways are taking on different ways of representing these movie frames from each other. Interestingly, when you compare to the one pathway model, what you see is, so here we're again doing representational similarity analysis on the neural networks, but this time we're comparing the two pathway network to the one pathway network. We find that the blue pathway is more similar to the one pathway network, whereas the red dorsal pathway is not similar. And this just verifies what we already kind of saw in the other analysis, that when you train with this one pathway network, you're getting more ventral-like representations. So the two pathway network has these two specialized pathways, but the one pathway network really just kind of takes on most of the functionality of the ventral-like pathway in the from the two pathway network.
Can I ask a question?
Yeah, sure.
So with these two pathways, do these different representations kind of just spontaneously emerge? Like you've not done, these two pathways are the same pretty much, right? Is that?
Yep. That's right. We put in 0 other biases to generate these two things. So I should say, to clarify, what determines which pathway becomes which is initialization. Yeah. And so we're doing multiple random seeds and initializing multiple times. And then in order to do these comparisons, we are always first determining, okay, which of the pathways is the more ventral or more dorsal-like, and then we run the analyses.
Right.
An interesting question that I'll return to is, what could you do to ensure that one of the pathways becomes the dorsal and one of the pathways becomes the ventral?
Sure.
So to further judge the functional specialization in these networks, we then also did some downstream tasks. So what we did is we took the representations learned with CPC and we froze the weights, and then we just trained a linear decoder to do one of two tasks. Either we did CFAR10 categorization, or we did motion discrimination. So we were using these things called random dot kinematograms, which is basically where there's a bunch of dots moving across the screen in a particular direction, but some percentage of the dots are moving in random directions, and you have to judge what is the principal direction of the ones that are moving together. And you can change the difficulty of this task by altering the percentage of dots that are moving coherently versus moving randomly. So it's again a categorization task of 4 different directions, but now you're not trying to categorize object identity, you're trying to categorize movement. And what we find is that if you train linear decoders off of these networks, you can get better performance on CFAR10 with the ventral-like representations, and much better performance on the motion discrimination task with dorsal-like representations. Now, it's worth saying that the CFAR10 accuracy is really low and not that much better than what you get with an untrained network. But of course, that's not crazy because these networks were trained in a self-supervised fashion off of UFC 101, and we didn't do any kind of optimization for the categorization task. This is just like no hyperparameter or anything, literally just take our frozen representations and train off of those. There would probably be ways to get that to work better given the results that people have had with other self-supervised techniques and doing linear decoding off of those. But the key thing that I want to emphasize here is just that we get better results off the ventral-like pathway, showing that in the ventral-like pathway, there's the sorts of features that are being extracted from the images are better for doing object categorization. And then likewise, for the motion discrimination task, where it's much more obvious, in fact, the representations in both an untrained and ventral-like model can't do any better than chance. So chance level is here in the dotted line. So you can't even do motion discrimination really better than chance with these pathways. But the dorsal-like pathway, you can get way better than chance, showing that the representations in that pathway are much, much better for actually doing that motion discrimination task of judging coherent motion. To understand that a little bit more, we also looked at what happens when you change the difficulty of the motion discrimination task. So as I mentioned, you can change the difficulty of the motion discrimination task by altering the number of dots that are moving in a coherent direction with one another. Obviously, it gets harder as you drop the number of dots that are moving in a coherent fashion. And what you find is that at 100% coherence, then you can get better than chance. I should say the last piece of data, this was with 50% coherence. So when you're at 100% coherence, you can do better than chance with the ventral-like pathway, but effectively that means that that suggests that it can't integrate motion kind of globally across the image. The only thing it can do is it has a few local motion detectors that it might be able to use to do this task to some extent, but it gets easily confused by any distractor objects. Whereas for the dorsal-like pathway, even at 50% coherence, as I showed, you're still doing pretty well, showing that it can ignore those, like the representations are sufficiently global with regards to motion that the incoherent dots can be ignored. And in fact, even at just 20% coherence, you're still doing better than chance with dorsal-like representations. Another interesting question for making the task more or less difficult is the number of frames that you present. So how long does it watch the motion? Because obviously, if you see a small clip of the motion of the dots, it's more difficult than if you see a longer clip. At least you think so. And indeed, that's what you see for the dorsal-like representations. As we increase the number of frames that you play, the accuracy on that gets better. This is for 50% coherence. Whereas interestingly, with the ventral-like with the ventral-like pathway, what you get is a drop as you increase the number of frames in your accuracy. And what's really odd about this is that this demonstrates that a big difference between these pathways is that the dorsal-like pathway has learned to integrate motion information over time. So it is really taking into account, okay, what was the motion I saw a few frames ago? Whereas the ventral-like pathway doesn't seem to be doing that at all. It can detect movement, but it's just doing it in a small time frame. And in fact, it makes it harder for it if you provide more time frames, because then it maybe gets a bit more confused. So this just further emphasizes the distinct kind of functional specialization that we see in these two pathways in a network trained with a single loss function.
Hi, could I ask a quick question?
Yeah, sure.
Were these networks trained on videos that you're testing with the random dots, or were they trained on the random dots?
No, they were trained on the UFC 101 data set,
right,
and then they were tested on the random dots.
So is there a sense that if the video progresses slowly, then you're quite good at prediction if you can say what, if you understand the current scene, but as the video moves more quickly, you need to be able to predict further?
Yeah, I suppose that that seems a reasonable hypothesis, yeah.
So if you had sort of faster moving videos that you trained on, you might see more representation of dorsal-like representations, even in a single network, perhaps.
Perhaps, although I have a control to speak to that.
Okay, thanks.
Thank you for the segue. So what's interesting though is if we take the same data set, the UFC 101 data set that we trained previously on, but now we just changed the loss function. So now we're doing supervised training. We're getting the network to categorize the different actions in the data set. Now we don't get good fits to the dorsal regions at all. We're back to a situation more akin to what we had with the one pathway model, even in the two pathway model. So we get some decent fits to some of the ventral areas, though less so Viz-PM, but for Viz-LM and Viz-P, we still get some decent fits, but the kind of dorsal-like representations get eliminated. And this is just now comparing across the kind of summary data for the groups. So again, for ventral regions, whether you're using one pathway or two pathways and you're training on action recognition, you get better than untrained network representational matches for the ventral regions, whereas for the most dorsal region, Viz-AM, you actually get worse than an untrained network, in fact, with the two pathway model by training on the action recognition. And so what this speaks to is that it's not just the statistics of the movies themselves that induce these distinct representations. It's something about the statistics of the movies and the particular loss function that you use. So it's something about the loss function itself that is also helping to encourage these distinct representations and these ways of integrating. Now, that being said, I'm sure that if you change the movement statistics in the movies, per your question, Neil, you could probably develop, again, kind of distinct representations. I'm sure you'd see differences in terms of what your two pathways produce. But what this does demonstrate is that it's not purely just the nature of the motion in the videos that is encouraging this stuff. It's also something about that loss function. And indeed, so this is just showing that when you now compare across the two pathways in the supervised training network, you no longer see as clear a distinction in terms of the representational similarity between the networks. So what this shows is that in the supervised training case, there doesn't seem to be a sort of competition between these different types of representations. There isn't something pushing the network to develop one set of representations versus the other. It can solve the task with just a single sort of representations. And this suggests that these two forms of representation are competing with one another. And we see that when we look at the correlation over training in what we call the V-score and the D-score for the network. So this is just, if you take the most ventral region, Viz-LM, or the most dorsal region, Viz-AM, and you measure the representational similarity, between your neural network, the max representational similarity for all the layers and these regions. So this is basically telling you how much is there a part of the network that matches the ventral pathway versus a part of the network that matches the dorsal pathway. In both networks, we see a negative correlation, but in the one pathway network, we see a very strong negative correlation of like negative 0.54. So meaning that as the network becomes more dorsal-like or more ventral-like in its representation, and I'm sorry, I think the colors got switched here on this diagram. I'll have to change that for the paper. But as the representations become more ventral or dorsal-like in the matches, the other type of representation drops in its similarity. So as the network becomes more ventral-like in one of its layers, it's necessarily becoming less dorsal-like at the same time. That negative correlation, though, drops for the two-pathway model. Not completely, it doesn't disappear altogether, but it does drop significantly. And you can actually, over training, see more clear increases in your ventral score and dorsal score simultaneously, suggesting that part of what's happened here is that the loss function, the predictive loss function, has competing interests. And this gets back to my original hypothesis. There's a competition between these two different forms of invariance, a sort of like identity invariance versus a movement invariance. And these two different types of invariance are competing with each other for doing the predictive task. And what happens when you have the two-pathway model is you allow the network to find a solution that resolves the competition a bit better than you can with a one-pathway model. And I think this is a key thing because, of course, to some extent, it's strange that we see these functional specializations in the two-pathway model, given that it's the same number of kernels. So like, why would this happen? But we think it's because in creating that separation, the gradient can end up, kind of finding out, oh, like I can shuffle these, I can send these representations off to this pathway and these representations off to this pathway, and they're not going to be mixing. And so it's going to keep them nice and separate so they don't compete with each other. So to wrap up, one of the things that's nice about this is our results match those of another recent study showing that self-supervised learning induces better matches to mouse visual cortex than supervised learning. This was from Dan Yemen's lab, just came out. And what you find is that ImageNet categorization accuracy, for example, does not produce a good prediction for how well you match mouse cortex. Instead, you get your best results from unsupervised or self-supervised contrastive losses, and also with relatively shallow networks, I should say. So in this particular study, we use deep networks, but one of the things that Dan's lab finds is that they get their best representational matches to mouse when you use shallower, so not completely shallow networks, but just a few conv layers rather than several. This is in distinction to primate cortex to some extent, because in primate cortex, you can see that you get good matches to ventral stream regions with supervised learning on ImageNet, suggesting that there's maybe some differences in loss function. That's also kind of highlighted by the fact that you see different abilities between rodents and primates in terms of their capabilities to do object detection. So this is a recent paper from Doris Sau's lab. So I'll just show you her stimuli here. We're going on to Twitter for this. So maybe you can see this or not. So here, this stimuli, I'm sure you can all see this object moving on the foreground to this background. So can other primates, but mice cannot. So there's something about the mouse visual cortex that is slightly different from our visual cortex. We don't fully understand those differences, but it is, I think, something that will be kind of uncovered a bit more over the next little while. Another thing I'll say, though, is that in order to fit to primate cortex, we have another paper out that's in review right now showing that you can get good fits to primate dorsal stream by doing actually a different sort of self-supervised task, which is where you take image sequences from a virtual reality and you basically predict the velocity of the agent. So the velocity in 3D plus rotational velocity and stuff, if you predict those velocities from the movies, then you get really, you get good matches to primate dorsal pathway. So it's possible that primates have some additional kind of like things that they're trying to do beyond just prediction, whereas mouse cortex is maybe concerned largely with prediction. I'm going to skip over that because we're running out of time. So to finally wrap up, these results demonstrate that we can explain specialized visual pathways in the mammalian brain, at least partially, using a predictive loss function and two distinct pathways. Some additional cues that we're interested in exploring. So this comes back to one of the questions asked. Can we find other inductive biases that guarantee which pathway takes on movement invariance and which takes on object invariance? So what could we do? Perhaps by adding that velocity prediction task, we could ensure that one of the pathways becomes the dorsal-like pathway. Another interesting question is, what about other self-supervised losses that exist? So if we did this with like Barlow twins or something like that, you know, would you see the same division of labor, bootstrap your own latent, etc.? Another question that I've already touched on is what combination of losses will produce the best fits to primate cortex? And in particular, how do you get better shape-based representations? Primate ventral stream seems very concerned with shape, and it's one thing that most of the existing neural network models don't successfully do as well so far. Lastly, where might these loss functions actually act in the real brain? Like, where are the predictions happening? Are they happening? Because as I noted with CPC, you're doing predictions in latent space. Possibly, you know, the prediction is happening up in the hippocampus, and then error signals get propagated down through the visual stream. Who knows? And the last thing I'll mention, per my promise, is another interesting thing I think about with regards to this data for engineering applications for artificial intelligence is this question of how do you learn good representations for control? So people have tried using kind of predict next frame pre-training to set up representations for doing control, like moving robotic arms and stuff like that. And it kind of works, but not great. You know, like I've spoken with Chelsea Finn about it, and they've done this kind of stuff out there in California, and they find that it's like decent, but not great. And one of the things that I wonder about is, per this finding that there seems to be a competition between these different types of invariances when you're doing predictive training, it's possible that actually to get a good set of representations for control, you want two pathways, because it seems like when you have one pathway, the predictive loss is best minimized by taking on ventral-like representations, which don't care about movement and do care about object identity. But if you want to do control, if you want to have a robotic arm that's interacting with objects or, you know, moving things around, etc., you probably want some representations that are also really good for capturing movement of objects in the environment. And so for that, I wonder if one of the missing ingredients in some of the AI pre-training with predictive loss functions to date has been that they're still training on a single pathway. And in fact, to achieve good representations for control, you need to resolve that competition. And it's interesting then to think also for the brain why we might have evolved separate pathways that way, because indeed our dorsal stream is what feeds into our control systems, our motor control systems and stuff like that. And it's possible that, you know, one of the things that encouraged this anatomical split is precisely that that then allowed for representations to emerge that were better for control. So that's that. Thanks very much for listening. I also want to thank Shahab for his fantastic work on this, and the folks at the Allen for their constant efforts to make rich neuroscience data sets available to everyone, and of course to our funding agencies. And thank you for listening.